let's talk about functional genomics. And we've talked about genomics before. Genomics is basically studying all the sequences to begin with, or to start with, of genes, obviously, but it goes beyond genes. It goes into sequences in DNA that may control other aspects of cells' life, like DNA replication, alignment of homologous chromosomes for meiosis, et cetera, telomeres, centromeres. So genomics is the study of all sequences in a cell's or a species or individual's DNA, right? Functional genomics is sort of an extension of that and is to try and guesstimate to the best of our abilities what a cell or organism might be able to do based on the sequences that we see in that DNA, right? So genomics is the study of all genes. Functional genomics is taking it just a step further and say, can I predict to some extent, to some degree, what a certain cell or individual might be doing based on the sequences that I see in their DNA. So before we dive deeper into functional genomics, I wanted to readdress something that we touched on last week when we talked about DNA sequencing. I mentioned that modern capillary sequencers, which we still use every day for short sequencing runs, will generate approximately 800 to 1,000 base pairs of sequence in about an hour or so. 40 minutes is net time, but give and take about an hour, right? The human genome is 3 billion base pairs long, so if you do the math, it would take approximately 7 plus years, if you were running only one sequencer 24/7 nonstop. We figured that was unrealistic, but that the approach that the world took was to simply throw many sequencers at the problem. And with hundreds of sequencers all around the world, mostly in the US though, you could theoretically sequence the human genome in a little over a week. Now, there was a problem with this reasoning. Right? So this reasoning is based on the idea that if I have close to 400 sequencers running simultaneously, in about a week I'll have the sequence for the human genome. But there is a problem with that argument. And that problem relates to a question that I wanted to ask of all of you. What is it that you need for Sanger sequencing or capillary sequencing? What is it that you need to obtain sequence from DNA using this technology? Does anyone remember from last week? What is it that we use?
I mean, isn't first you need dyes for it?
Dyes?
To color like the different lengths.
All right, so those dyes, there were not only dyes, right? What else, what other feature did those things have? What was special about them biochemically? That's right. So we need the DDNTPs. Well, that's not very thick, is it? The DDNTPs, each of them color one way and in partial abundance, right? So that there would be regular NTPs as well to let the DNA synthesis progress in most of the runs. What else? So the DNTPs, fluorescently labeled. What else do you need? The DNA itself, the DNA that you want to sequence, right? Thank you. Yes, the template DNA. And now that everybody shook it off a little, what else? The primers. The primers. And that's where things get a little tricky. I'm going to do it here in bold letters. Why do things get tricky there? Because what do you need to design the primers? You need to know a little bit about the sequence that you want to sequence. So it's one of these, what's first, the chicken or the egg problems, right? We need some information about the sequence that we'd like to sequence so that we can design a primer that would allow us to sequence more. right? Sanger sequencing, and by extension, modern capillary sequencing, you always need some sort of starting point. So you can take a DNA sample, just any sample, let's say not a human genome, but you find a new species of fungus in the forest, you'd like to sequence its genome, you have no idea about any of its sequences. Where do you start? How do you design those primers? You might say, well, I can see this fungus, I recognize that it probably belongs to this or that other family. I'm just gonna throw in some primers against very conserved ancestral genes that this fungus probably shares with many other fungi. So I'm gonna use those primers just to get me started. And then you start. But what do you do with everything else, right? So you always need a little sequence or knowledge of the sequence in order so that you can design the primer so that you can continue the sequence. And that's what threw a wrench at simply saying, we'll just use hundreds of machines and sequence all at once simultaneously, and that's gonna get us over the hump in a week. So in fact, what delays the process of sequencing using these type of technologies is that, like I said, you need to sequence some so that you can design the next primer, so that you can sequence some more so you can design the next primer, and so forth. That's illustrated down here. And this is a process that we call, rather intuitively, genome walking. All right, so you sequence some, you retrieve that sequence, you design the next primer near the end of the sequence that you just obtained so that you can walk some more. Then you retrieve that second sequence, design a primer near the end of that new sequence, and so forth. So genome walking. Because there is that retrieving the sequence, designing a primer that will bind to the end of it, that will be relatively specific, that we won't stick to other things, that it won't stick to itself, and so forth. Now, we go from, for every 800 to 1,000 base pairs, instead of taking somewhere between 40 minutes and an hour, it'll take a whole day, because once you've decided on the primer sequence, you need to synthesize that thing, and that takes a few hours too, and purify it and get it, and so forth, right? So it added quite a few hours to every little bit of sequence that was being generated. So now the math changes a bit, and instead of dividing 3 billion by, let's say, 800, and turn that into minutes, it turns into days, and now 7+ years becomes over 10,000 years, and about 26 years if you're running close to 400 machines at once, right? So from the beginning, we knew, I say we as a proverbial humans knew, that it was gonna be a problem to sequence any genome, let alone the human genome, doing genome walking. It just takes a long time, right? So people immediately started thinking of alternatives, and the idea that immediately came about was the idea of parallel processing. So instead of generating sequences sequentially, one after the other, based on information that you obtained before, you just parallel sequence different regions of the genome. So you generate all these random sequences from everywhere in the genome, and then you turn it into a puzzle. You just need to assemble these sequences into what may most likely be a continuous sequence that you hope will resemble the original one. We call that shotgun sequencing because you have your genome that you like to sequence, and it's almost like you're shooting it with a shotgun. And pellets are going to spread everywhere and hit everywhere in this genome. Same thing. So here, you're throwing at this thing a whole bunch of random primers, and I'll explain in a second what that means, such that you start generating brief sequences from all over the genome, sometimes in a way that these sequences will overlap one another. And that's actually something you want by design, because it is that overlap between all the sequences that you get back that will allow you to reassemble the sequence, right? That put together that puzzle, if you will. Okay? A bit of technical terminology, all of these different fragments that get assembled into a sequence. These assembled sequences we call contigs. So you may find to this day references to contig and a number. That's simply an indication that there may be entire chunks of sequence that you're working with. Perhaps not a human sequence, because now we have a pretty good annotated human genome, but let's say that you discover a new species, or you're simply sequencing from scratch a species that has been known but whose sequence has not been determined yet. At first, you may generate these fragments of sequence that you will call contigs. And it just comes from contiguous sequences. We're not going to go into a ton of detail into how you assemble sequences, but we'll just touch on the intuitive approach to it. And it's rather simple. Let's say that you have a sequence, a whole bunch of letters that ends in A. And then you have another sequence, say that you have another sequence that starts with A. So how confident would you be in stitching these two sequences together and saying, well, clearly that A was the one in between the two. Now I can stick them together into a contig. Not very confident, right? Because there is a very high chance that an A occurs anywhere in the genome. So how do you know that that is the one A of the overlap? You wouldn't be very confident because A happens 1 every 4 nucleotides in DNA. So the chances of finding an A are 25%. So I wouldn't bet that these things go together on a 25% chance that I may be joining them mistakenly, right? So it's too much of a high risk. But you can probably appreciate where this goes. When now you have a sequence that ends on A, C, C, G, T, A, C, T, G, A. And then you have another sequence that starts with A, C, C, G, T, A, C, T, G, A. You can ask yourself the question, what are the chances that I might find these two sequences in two separate places in the genome and they actually don't go together? And the math that you will run, it's a math that we're familiar with. You will do 1/4 times the number of positions that you're looking at, right? So that is the probability, the combined probability that that particular sequence happens twice in the genome just by chance. I made it sound super simple, right? And when you do the math, you may find that that is an unlikely low number, but let's not forget that the genome is an unlikely large sample too, or an unlikely large pool of sequences, right? So there is some math behind all of this. And there is also the help of sometimes these fragments that you're trying to unite together have already in them some sequence that you know of, because before DNA sequencing large scale came about, there was that very painstaking, laborious Sanger sequencing, people working with long gels. So it's not like we knew nothing about the human or the E. coli or the rat or the mouse genome before all of this started. We had some landmarks throughout the genome that had been put together through a combination of genetic mapping since the early 1900s to some individual sequences generated by Sanger sequencing since the early 70s. Okay, so there was some references, and those sometimes could help. Anyway, the point is that through overlapping sequences, where you would find a really nice stretch of letters at the end of one fragment that was identical to a nice stretch of letters at the beginning of another, now you could stitch them together like you would with two overlapping sections of a picture or text or whatever. So when you do that, and when people did this, in practice was like putting together a puzzle. And what happened very, very often is that they would generate certain regions of sequence or sequence from certain regions more than once. And then there would be just as likely, or perhaps even more likely, certain regions that were missing. Right? So imagine that you're trying to put together this puzzle with this very nice picture of a landscape. There will be some pictures that are kind of repeated. And that was by the very nature of shotgun sequencing approach. You throw these random primers at your target sequence. There may be some areas of those target sequences that get sequenced twice or 3 times or more. Right? And just by luck, there may be certain areas that don't get primed at all. So there is that aspect. right? The sort of the stochasticity of throwing random primers at a target sequence. And then there's another aspect, and that is those random primers that scientists were throwing at their target genomes had some complexity to them. Right? So you can put together a primer that is 4 or 5 nucleotides in length, such that there is some chance that it will bind to DNA and allow the initiation of DNA synthesis and the sequencing reaction. But there are entire areas of all DNAs, not only human DNA, of all DNAs, maybe a little bit less so with prokaryotic genomes, but eukaryotic genomes, they're full of them, of and so forth, right? There are entire regions that are like that. So unless some of your random primers were TTTTTT, those primers would never, or your random primers would never land on these repetitive sequence regions. Does that make sense? So sequencing effort after sequencing effort, scientists encountered this problem that there would be gaps in their sequence. They ended up with collections of contigs, some of which contain sequences that they knew, so they could assign certain contigs to certain chromosomes, right? But for the most part, the first approach to any new genome sequence was just a library of contigs and then gaps in between them that were unknown. So, chromosome walking, very, very accurate, but it takes forever, and it allows you to put the picture together in essentially one go, but it will take you 26 years to do it. Shotgun sequencing will generate a collection of contigs with gaps between them that by definition you don't know. So knowing this, when the Human Genome Project got started, they decided that they would take a combined approach. So there would be shotgun sequencing for generating as many contigs as we could, and then there would be chromosome walking for the areas that represented gaps. So the idea behind that is people would go to the very end of a contig, design the primers that would allow them to sequence from there on, and then very laboriously and painstakingly would go from there. And the reason I say laboriously and painstakingly is because those gaps, like I said before, happen often for a reason, because these are areas of genomes that are very low complexity, very hard to sequence. It's just a string of A's and T's at times, or like nothing but C's at others. Right? So it's very difficult to sequence those things, because what happens when DNA polymerases run into repetitive sequence in their template DNA? They start making these mistakes of slipping forward, slipping back, so you're not quite sure if what you're counting is accurate or not. You have to do it many times to make sure the sequence you're getting back is accurate, blah, blah, blah. So a combination of shotgun sequencing and chromosome walking to try and fill in the ends. These people had a whole bunch of data that allowed them to kind of estimate how long it might take to do the whole human genome based on prior experiences, and they estimated that they would finish in 15 years. They finished in 13. Not bad. One last thing to mention is that we went from a little over a week with 400 machines to 26 years if we thought, or if we consider chromosome walking as the approach, right? With 400 machines or so, we're still back to about a week, except that obviously filling in those gaps took forever, but we're back to a week if those machines were running nonstop, like, no rest whatsoever, which is silly. Because what each of those machines would be doing is generating one of these fragments. It's just that they would do it simultaneously. So instead of waiting a whole day until you can generate the next stretch of sequencing, they could do it simultaneously. So within 40 minutes or so, you had about 1,000 base pair of sequence. Clean the machine, change the tubes, kind of wipe it up a little, then you do it again. And in a day, you could generate several thousands of base pairs of sequence. Okay? With that, they still calculated that it would take a while. All of that until next generation sequencing or modern sequencing. And again, I stressed this last week. We still call it modern, but it's already 15 years old or so. But next generation sequencing came along, and now we're not talking thousands of sequences being generated simultaneously in a given day. We're talking thousands of millions, billions of sequences being generated simultaneously in a given day, right? And we went through next generation sequencing using Illumina's approach as an example. There are others. They all conceptually, they share some things, but we went through the Illumina approach, and super briefly, the idea was that first you generate many, many copies of each of the random fragments that you would like to sequence through that bridge PCR for generating the clusters. And then for each of the clusters, you go through this iterative loop of adding nucleotides and polymerase. These nucleotides are special, so only one gets added. They can wash all of the other nucleotides and the polymerase away. They can shine light on this thing and basically ask the question, what are you? Right? Because each of these nucleotides would fluoresce a given color. Then once you know what they are, you can send in chemicals and an enzyme that will unpause the reaction by restoring that 3' hydroxyl that DNA polymerase would need to add the next nucleotide. So you send the chemical and the enzymes, you let them take care of that and kind of unblock that nucleotide for the next reaction. You wash them away, then you bring in the nucleotides and polymerase again, and then you repeat the process over and over again. And you do it several tens of times. You don't do this hundreds or thousands of times. In fact, the sequences read by next generation sequencing are typically somewhere between 50 and 80 or 100 nucleotides in length. But because you're doing it in such a massively parallel manner, you can generate millions of 50 base pair fragments at once versus a few hundred, 1,000 base pair fragments. Okay? One thing that next generation sequencing allowed is the volume of information that you get back from these things is such that sometimes just the sheer number of puzzle pieces that you have to put together might allow you to actually, with very sophisticated math on your side, might allow you to put together longer contexts. So next generation sequencing sometimes might lead to the assembly of almost complete chromosomes all at once, because it's just the sheer number of sequences you can afford playing the numbers game. And if you find, let's say, a short region of overlap between fragments A and B, and then a short region of overlap between fragments B and C, and then you find even some sequences that overlap A to C, and you can confirm that B is in between, slowly but surely you gain confidence that you can start stitching these things together, and your confidence in your final sequence increases. So it's just the sheer volume of information that you get back allows you to statistically or probabilistically be a lot more confident about your contig formation. Okay? So it is rare nowadays that you would send a sequence out, or you would send a sample out for next generation sequencing, and that you would get back a collection of hundreds, if not thousands of contigs and a whole bunch of gaps that you need to resolve by yourself. Typically, you will get back entire chromosomes assembled. And of course, you always can contrast that information to whatever you know about your species of interest separately. So if we're talking a prokaryote, we'd probably know that we're expecting some sort of circular chromosome, back some circular sequence. If you're talking about a eukaryote, it might take just a bunch of relatively simple laboratory tests to find out what the karyotype of those individuals are. There are really old techniques that allow you to take cells from an individual and force them to form the metaphasic chromosomes and ask the question, how many chromosomes does this species have, and so forth, right? No? Okay. The reason we went through these historical hurdles that we had to go through with all DNA sequencing techniques, in what at the time was a very ambitious project like sequencing the human genome, is mostly so that you can pair up that bit of history, which ultimately, it's interesting, but it's not that important, with the biological basis for all that, why it is that it was so hard to assemble these sequences. And once again, the main culprit was these entire regions of repetitive sequence that are very, very hard to sequence by capillary sequencing. Okay? All right. So let's say that you have, say, new species or new strain, or the meteorite hits Earth and you find a bacterium on its surface, and you want to sequence their genome, and you get the sequence back. And now what? So the sequence that you get back doesn't look much different than this. So what do you do with that? What do you do with that? So what you do with that is something we call genome annotation. And what that means is almost like, when you do your annotated bibliographies, or you used to do your annotated bibliographies, where you go into each reference that you use and you explain a little bit of what that is. Not much different here. So you go into the sequence and you look for things that have some meaning inside of cells when they're written in DNA, right? So certain sequences that carry information in them. So my question to you, and now we're going to take a couple of minutes. What sequences could you look for here? The Tata box, okay, or GC region. You look for specific sequences that could make proteins that in a way right? So the sequence of certain proteins. You set kinases, so these proteins that phosphorylate other proteins or other molecules. You could look for those, okay? So we're going to be looking for, I'm going to just call that specific proteins. And how would you do that, by the way? The amino acid sequence, right? Typically, of a protein, what we can know is the amino acid sequence. So how do you guesstimate what the nucleotide sequence that codes for that amino acid sequence might be in a genome? Based on codons, you use the genetic codes to sort of, on paper, reverse translate that sequence of amino acids. So if you know that your kinase is alanine, glycine, cysteine, you look for the genetic code, and now you know that there is a specific combination of codons that could give you that alanine, glycine, cysteine, I think I said, right? Now, how many codons code for each amino acid? 4, 2, right? Sometimes it is 2, only once is 1, there is 2, there is 4, right? So if you have 3 amino acids and each of them could be coded by 4 different codons, you have a 4 times 4 times 4, 64 different combinations. Ouch. So are you going to be looking for 64 different words in this sequence? No, the trick is that you know that those 4 codons that code for the same amino acid share the same the first 2 letters. So you don't have to worry about that third letter. That's the one that differs between the different codons. So instead of looking for a word of 9 letters, you look for a word that has 2 letters, a gap, 2 letters, a gap, 2 letters, and a gap. Right? You can't do that easily with Microsoft Word, but you can code a computer to do that for you. Right? So all of these tricks happen. We can do those things. So we can look for specific protein sequences and say, this is my new fungus sequence. I'd like to know if this fungus codes for the protein that I know of in that other fungus. So I'm going to reverse translate that protein that I'm interested in, and I'm going to look for sequence, and I'm going to write a computer code that says, look for a string of nucleotides that could give these sequences of amino acids. Yeah. Cool. So what else could we look for? Terminator sequence. And here we get into a pickle. So When you say terminator, what do we immediately think of? Prokaryotes or eukaryotes? Prokaryotes, right? Terminator sequences are present in protein coding genes of bacteria, prokaryotes. When we think Tata box and GC-rich regions, what are we thinking? Eukaryotes, right? Both are possibilities. You just need to know what sequence you're working with. If you know you're working with a eukaryote, you would be looking for terminators. If you know that you're working with a new prokaryotic species, you would probably look for terminators. Would you look for a Tata box and a GC-rich region in a prokaryote? I see some heads nodding. What would you look for? What's the equivalent of the Tata box and the GC-rich in bacteria? Louder? Close. So Scheindel-Gardner was in the mRNA to allow ribosome binding. And yes, we would look for those. That's right, the minus 10, minus 35. That's right. So minus 10, minus 35. And why not? That's sloppy. Come on, I can do better. And a good Scheindel-Gardner is what you would look for in bacteria. Right? What else could you look for? So these two, the Tata box, GC-rich, the minus 10, minus 35 combo, and a strong Scheindel-Gardner, say promoter in prokaryotes and eukaryotes. Specific proteins we look for already, terminator. What's the equivalent of sorts of a terminator in eukaryotes? For sure. So you could look for the terminators that are important for.
Isn't the row dependent independently for the RNA when it cuts it out?
That's right. But you could still look for those. You're annotating a new eukaryotic genome, and those things might tell you where those genes are. So that's still important. But in protein coding genes in eukaryotes, what's the sort of a functional equivalent of the terminator that says to the cell, yeah, this mRNA should end here? The poly-A. Now, the poly-A is not written in DNA. You will never find T that gets transcribed into A. No, but there is a poly-A signal that says to the poly-A machinery, you should cut this primary RNA here and start adding some A's. That's based on a signal that we call the polyethylylation signal.
Does it fit in the specific proteins that you're looking for?
You can make the safe assumption that they will all have it, because that's a generic feature of protein coding genes in eukaryotes. So all mRNAs will need one so that they can have a poly-A, so that they can get translated and so forth. So it doesn't apply to specific genes. I mean, it applies to specific genes in the sense that it applies to protein coding genes. Not all genes are protein coding, but among protein coding genes, they will all have it. I heard AUG, right? So before you propose the idea of maybe looking for genes that we know from other species, but what if I wanted to see what new genes does this thing have? I just found this mushroom in the forest. I'd like to see what new proteins it makes, right? So how do you go about looking for new genes? And I heard some of you say AUG. AUG is a good start, no pun intended. It's a good start. But what else would you look for when you're looking for new protein coding genes? Well, you would probably look for a combination of a series of things. You would hope that the AUG somewhere upstream of it has a strong promoter. You would hope that that AUG may have upstream and downstream of it some splicing donor and acceptor signals that you know are going to be used by the cell to splice the mRNA where those AUGs are. And more importantly, you would hope that that AUG is followed by many codons that are not stopped. Because if you find an AUG, we can do the exercise, we can find an AUG here easy. There's an AUG. Oh, that's too thick. But I swear I found one. ATG transcribes to AUG. If I take that AUG and I follow it, and 3-4 positions later I find a stop codon, that was not a protein. Right? So that approach of sort of homing in on an AUG and see how far we can go and how complex that sequence might be, because AUG, 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 AUG is not very meaningful either. So we want an AUG followed by complex sequence that goes for long until we hit a stop codon. Those are things that we call open reading frames. ORFs, open reading frames. And that's one of the first approaches that we take when annotating a new genome sequence that we know nothing about. So we scan the genome for entire areas that may have a collection of what might potentially be a string of codons that codes for a string of amino acids. Then if we can pair up strong promoters upstream of them, in the case of prokaryotes, strong terminator signals downstream, and in case of eukaryotes, some strong poly-A signal, then those are pieces of evidence that start piling up and that say, this may be a gene. This may be a gene. And at that point, we call that a gene model. So anytime that we have a new genome and we start annotating it, there will be many gene models. Then we're also going to take the approach they were proposing before. We're going to take known sequences from related species. We look for those in this new genome, and there's a good chance that we'll find them. After all, we share sequence with all the way down to bacteria, right? So we will find sequences that are conserved across phyla, and the closer to specific species are, the more of those conserved sequences you might find. Right? So it's going to be a game of I look for known knowns. So I look for sequences that I know of in other organisms. I find them. So I have pretty high confidence that these are real. Then I'm going to look for some unknowns as well. So you will look for these open reading frames and hope to come up with a bunch of gene models that at least you can investigate further, right? I want to make sure that we get through all of it. We have about 10 minutes, so we should do fine. So genome annotation is mostly gene predictions, these gene models, and then some other DNA features. We call them DNA features. Things like centromeres and telomeres and regions that we know are usually, say, insulators or known enhancers and so forth, right? So other DNA features as well. Once we've generated gene predictions, then we can start designing transcriptomics tools. So let's take a second to think about that name, transcriptomics. Now we're studying the collection of all RNAs present in a sample. We took that sample originally, we extracted all the DNA in it, we sequenced its genome, so we did genomics. But now we can design transcriptomics tools that allow us to look at their RNAs. And those are going to come in two forms. Microarrays, they're still produced, they're no longer created. They became a deprecated technology, but companies will still make microarrays because the technology is still useful and it's cheap. So you can still buy microarrays. Most people these days do RNA sequencing. We'll go through microarrays first for two reasons. The literature is flooded with studies that used microarrays, so there's a good chance if you engage in scientific literature at some point that you may bump into them. And then, of course, we'll talk about RNA sequencing. And the other reason why we talk about microarrays is because it sort of lays the conceptual ground for understanding RNA sequencing. So in microarrays, the idea is as follows. We have all of those gene predictions, right? All of those gene models. We're simply taking a bet. We're saying, I think there may be a gene here because I see a sequence of nucleotides that start at an AUG and go for many codons before we hit a stop codon. So I think there is a gene here. So what we do is we print all of those sequences onto a special glass slide. We use these very fine robots that go and literally deposit each of the nucleotides from those gene models. So on the glass slide, you have all of these spots, again, not millions, like in next generation sequencing. These are in the hundreds or the thousands. On these class slides, you have all of these spots, and in each of them, there is a certain group of words written in them. And when I say a group of words, I mean the same word written several times, right? And each of those words correspond to these gene predictions that you made. So you have that. Then separately, you take your sample, and typically microarrays are done between some sort of control and an experimental sample, right? So it could be and the reason for this is as follows. Genomic information is important for trying to determine what's there, right? You're trying to predict what genes a given species may have. Transcriptomics is more about how is the species using this information. Looking at RNAs means looking at what those species are expressing at any given time. And that's something that matters more to us when we're asking questions about the use of this information, not so much the having the information, right? So transcriptomics, typically, you have a sort of a control design. You have a control and an experimental sample. And what you do is you extract RNA from both, and then you mix those RNA samples with a reverse transcriptase, and then dNTPs, not dideoxy, just regular dNTPs that are labeled with a given color, a given fluorescent color. We are going to use red fluorescence for our control, and then green fluorescence for the experimental. That reverse transcriptase will take all of the RNAs present in each sample and will reverse transcribe those RNAs to cDNAs, to copy DNAs. And because that reverse transcriptase had red building blocks in one tube and green building blocks in another, what you end up with is two tubes. The experimental one has cDNAs that fluoresce green, and the red has cDNAs that fluoresce red. And the control, I mean. The control has cDNAs that fluoresce red. Then you take these two tubes and literally dump them on top of that slide. And these cDNAs labeled red or green will float around until they find on the slide a printed sequence that is complementary to them. Right? Think about the mechanics of this. We had an RNA that has the same sequence that is present on the slide. When you reverse transcribe using fluorescent cDNAs, you're going to generate the complementary sequence. That complementary sequence is complementary to the RNA they came from, and it will also be complementary to the sequences found on those glass slides. Right? So you make a complementary sequence in the tube, and then you use that complementary sequence to probe these printed oligonucleotides on the slide. And then there can be a number of situations. If a given gene is not expressed in either sample, because it's just not expressed, it's off, or because you predicted a gene that doesn't really exist, then neither the green probe nor the red probe are going to bind to that particular spot on the slide, and you'll see no fluorescence. That spot will be dark. If it turns out that your experimental sample expressed a lot of one gene, but that gene was not expressed in your control, you will see a lot of green probe binding to a specific spot, or a spot that corresponds to that gene, but very little to no red probe. When you look at the slide, that particular position will fluoresce green. We say that that gene is upregulated in your experimental sample. It is expressed more in your experimental sample. If the gene was expressed more in the control than in your experimental, the opposite. There will be a lot of red probe that finds the corresponding spot on the slide, very little to no green probe that finds that spot. When you look at the slide using a camera, you will see that that spot fluoresces red. And that means that that particular gene was down-regulated in your experimental compared to your control. It was expressed less than in your control. Do you guys follow that? The up-regulated, down-regulated? Yeah. And last, whenever you see a yellow spot, it means that about an equal amount of red and green probe bound to that particular spot, and that is simply telling you that that gene was expressed equally in both samples, in your control and your experimental, right? So expressed in both. So doing this, for years, people gathered pictures that looked exactly like this. That's a real picture. That's what this thing is pretty. It looks under a microscope. And they knew the identity of the sequences in each of these spots because human beings had printed these sequences in these slides anyway. So we knew exactly what's in each of these spots. And just by looking at the intensity of the fluorescence and the color in these spots, we could determine if a gene was being expressed or not, based on whether there is any fluorescence coming from a spot. And if there was some fluorescence coming from the spot, is it expressed both samples equal levels? Is it expressed more in one or the other, or vice versa? Does that make sense? You may, I hope you get familiar with the concept behind all of these, because I know for a fact that there may be questions in the test that say you label your control with red, experimental with green, now you see a yellow spot. What does that mean? You need to be able to answer that. That's microarrays, and for probably over a decade or so, a couple of decades, we collected transcriptomics data. So data about all of the RNAs being expressed by a sample, an organ, an individual, a given individual under certain conditions, right? So transcriptomics data we obtained from microarrays for decades. With the advent of next generation sequencing came something called RNA sequencing. It is important that you guys understand that it is not that we're sequencing the RNA directly from the sample, like in RT-PCR and like in microarrays. We first take the RNAs from both samples, we reverse transcribe them. We are going to obtain cDNAs from sample number 1, labeled green, cDNAs sampled from sample number 2, labeled in red. The difference is that these samples are actually not labeled fluorescently. We maintain that color coding to distinguish between the two samples. The NTPs that you use in these reverse transcription reactions are not fluorescently labeled. They're just regular DNTPs. It's just that I maintain the color coding so that you guys understand that one bunch of cDNAs is coming from one sample and one bunch of cDNAs is coming from the other. But they're all gray. There's no color in them, okay? Or transparent. That's probably more like it. What happens is now you take these cDNAs and you use next generation sequencing to sequence them all. Right? And before I told you that with next generation sequencing, you can sequence millions of nucleotides in a day. When we're talking about entire transcriptomes, that's a lot less than millions. Let's say the human genome has roughly 30,000 genes. Only about 20,000 of them express at any given time. And they may be, on average, 1,000 nucleotides long. So 20,000 times 1,000 nucleotides long, maybe 10,000. So you do the math, and it's about 200,000 or 2 million. So it's something that new next generation sequencing can handle just fine. Okay? So you do next generation sequencing, and then the next step is the step that is not super intuitive. So we'll take a minute to go through it. You have your two collections of reads. Once again, you can't see them. They're transparent. You just have sequences of letters in a computer. But then you ask that computer, hey, I know the sequence of the genome where these RNAs came from. You need to know that in order to do RNA sequencing. So I know the sequence of the genome that these things came from. Would you do me a favor and map these reads to different regions of the genome that we know? So what the computer will do is we'll take each of these fragments and say, oh, that maps to gene 3, all of them. These guys here map to gene 2. I also see a bunch of reads from sample number 1 that map to gene 2. And then I see some reads from sample number 1 that map to gene 1, but I don't see any from experimental sample 2. So essentially, the computer is telling you, hey, I see reads for experimental sample number 1 from genes 1 and 2, and for experimental sample number 2, I see reads that map to gene 2 and gene 3. And then you just use logic to say, okay, what that means is that experimental sample 1 is expressing genes 1 and 2. That's why I see cDNAs that came from RNAs that map to those genes. So these genes were transcribed, reverse transcribed, and then they map back to them. And I see that experimental 2, or sample number 2, expresses genes 2 and 3, not 1. And from there, it's almost like we did with microarrays. Some genes will be expressed by both. Some genes will be expressed by either of them. Some genes will be expressed by neither of them. And we obtain all of that information by mapping the sequences to, here it says ref-seq. That's a common term that we use, and it means reference sequence. It's just a short name of that reference genome that we're mapping these reads back to.